Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about a very important topic of biology or biotechnology, that is genetically modified organisms. So what is genetically modified organisms? So these are living organisms whose genetic material has been artificially altered or modified using genetic engineering technology. So basically this process involves insertion, modification of a particular or specific gene. Okay, and this whole process is different from that of your traditional breeding method. Okay, so this is how an organism can be modified genetically and then can be exploited for our benefits. Okay, so genetically modified organism can include plants as well as animals or any microbes. Okay, so genetically modified organisms are living organisms whose genetic material has been altered through genetic engineering technology. This involves modifying DNA of an organism in this way that does not occur naturally. So this whole setup is prepared artificially for our benefit. Genetically modified organisms or GMOs are used across various fields including agriculture for higher yield, in the field of medicine to produce a particular protein or hormone or any chemical which can be used as antibiotic or to treat any kind of disease. Next, we can use them in our research. Next, then what is GEAC, Genetic Engineering Appraisal Committee. So basically GEAC functions in the Ministry of Environment, Forest and Climate Change. It is responsible for appraisal of activities involving large scale use of hazardous microorganisms and recombinants. What is recombinants? The organisms whose genetic material is altered through recombination with other genetic material in research and industrial production from environmental angle. This committee is also responsible for appraisal of proposal relating to release of any genetically engineered organism or products into the environment, including experimental field trials. So this department is controlling all these things related to genetically modified organisms and their products, about their field trials before the marketing. Next, what are the gene editing tools? Using those gene editing tools we can modify or alter the gene. Okay, the first one is recombinant DNA technology. It is the most common technique to modify the gene. Next one is CRISPR-Cas9 technology. This system is a revolutionary gene editing tool that allows scientists to precisely target a specific gene, modify that particular gene. It can be used to add or delete or replace gene in a wide range of organisms, from bacteria to plant, from animal to plant. So this is another technique. Next is TALENs. What is TALENs? Transcription activator like effector nuclease. TALENs are another gene editing technique that can be programmed to target specific DNA sequence. They work similarly to CRISPR-Cas9 and have been used for genetic modification in various organisms. Next is RNA interference. So this technique, RNA interference or RNAi, is a natural cellular process that plays a crucial role in regulating gene expression within eukaryotic cells. This triggers the degradation of target gene's messenger RNA and results in reduced expression of any harmful gene of corresponding protein. Okay, next Next, we can use somatic cell nuclear transfer technique, which involves transfer of the nucleus of a somatic cell into egg cell from which the nucleus have been removed. This process creates a genetically identified organism. Dolly, the sheep, was famously created using somatic cell nuclear transfer. Next, we can use synthetic biology, which involves designing or construction of new biological entities, any device or part. Next, viral vectors, we can use viruses which can carry specific genes to target cells. They can be used in gene therapy to deliver therapeutic genes to treat genetic disorders. Next, selectable markers and reporter genes. These are genes introduced alongside the desired genes to assist the identification and selection of genetically modified organisms. So, selectable markers confer resistance to specific antibiotic or chemicals, while reporter genes produce easily detectable proteins, any fluorescent protein, to indicate successful gene transfer. Next, we can also use agrobacterium mediated gene transformation technique. Next, micro injection technique, which involves use of a fine needle to directly inject the foreign DNA into the target cell. Next, electroporation. The cells are exposed to an electric field which temporarily disrupt the cell membrane and allows the foreign DNA to enter into the cell. Next, there are some famous examples of genetically modified organisms. Here I am only giving you the examples of plants, genetically modified plants. The first one is golden rice, which is modified to produce higher level of beta carotene, which is the precursor of vitamin A. Next, drought resistant crops. Many drought resistant crops have been produced, which can the plants can conserve water to withstand the dehydration state. Okay, next is insect resistant plants like bt, brinjal. Bt means Bacillus thuringiensis, brinjal, Bacillus thuringiensis cotton. bt means Bacillus thuringiensis. The gene which is responsible for insect resistant is taken from the Bacillus thuringiensis bacterium, which is a soil bacterium, and it is inserted in these plants, which can make the plant insect resistant to overcome the infection by bollworm. Okay, so we can produce insect resistant brinjal, insect resistant cotton plant. Okay, next, papaya ring spot virus, papaya, genetically modified papaya, ring spot virus resistant papaya. We can produce flower sour tomato, was one of the first genetically modified food. It was engineered to have longer shelf life. Next, resistant cassava, a staple crop in many parts of the world, has been modified to resist viral diseases. Frost tolerant strawberries. These strawberries have been genetically modified to tolerate frost, allowing for extended growing season in colder climate. Non-browning apples. Apples have been engineered to resist browning when sliced or bruised, which can help to reduce food waste and increase their shelf life. Next, I have already discussed about GAC. Next, remember one thing about Cartagena protocol on biosafety. So it is primarily deals with the transboundary movement of living modified organism. It includes provisions related to handling, transportation and use of genetically modified organism, which can include GMOs in animals. An international agreement. It is an international agreement which aims to ensure the safe handling, transportation and use of living modified organism resulting from modern biotechnology. So remember Cartagena protocol and GEAC. Okay. So genetically modified organism offers some advantages like increased yield, reduced pesticide use, improved nutritional content, but also pose some disadvantages, which include potential health risks, then biodiversity loss, economic concern. So these are the pros and cons of genetically modified organisms. So it is all about genetically modified organisms. In a very short time, I have tried to discuss everything about GMOs or genetically modified organisms, and this video will be helpful to you. And if you liked it, please hit the like button, please share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.